Alright guys, welcome back. So the next category of macromolecules we're going to be discussing are proteins today. So in terms of where do you get these in your diet, well, they come from a pretty wide variety of foods, things like most people would think about, the meats. So your ribs, your steaks, your burgers, right, poultry, that sort of thing. But you also get a decent amount of protein out of things like eggs and nuts. Beans as well. Alright. So, in terms of functions, though, for your body, this is probably the widest, most varied functional category of all your macromolecules. Your proteins do insane amounts of things in terms of making your body or the body of other organisms function on a day-to-day basis. So, just some examples of functions and where they come from. Proteins can actually be used for storage. We can use it as energy storage. And a great example of that is the albumin or egg whites. Alright, so when you're eating those eggs, where's all the protein coming from? Well, it's coming from the egg white itself. Transport. Okay. Hemoglobin in your blood is a major protein, and its whole job is to be part of your red blood cells. And those red blood cells, their major function is to transport oxygen from your lungs to the cells of your body so that they have the oxygen they need to carry out other cellular processes. A lot of proteins have regulatory functions, so things like hormones, estrogen, testosterone, adrenaline, insulin, these are all protein molecules that help to ensure that your cells are communicating with each other and regulating certain processes so you're not doing too much of one thing or not enough of another to kind of keep you at homeostasis. Most kids, if I talk proteins and they have any background, the first thing they think of is their muscles. Alright? And in fact, most of the protein that the majority of us eat on a regular basis is meat from animals, and you are eating their muscle. Right? So two major proteins that are part of that are these fibers that make up the protein called actin and myosin. So those are both major protein structures. Proteins can be structural themselves. They're going to be part of our cell membrane structure, and we're going to talk about their importance later this year. But you've also got proteins that make up your hair, your nails, keratin, if you've ever come across that one. Those are some major classes of proteins in those sorts of structures. Disease prevention. Right? You've got an immune system that's job it is to keep you from getting sick from the same thing every time you get exposed to it. And that's because your body produces proteins called antibodies that are able to recognize those molecules and try to take care of them before they really do make you sick. And then you've got your enzymes, so things like lactase, sucrase, amylase, and these guys are going to be important when we start talking about how they influence your metabolism. Either they get involved with your chemical reactions, and in fact that'll be our last topic for this particular unit, is diving into exactly how enzymes work. So for now, keep in mind that enzymes are proteins, and that's going to become important for us later. So, you've got a whole list of functions to keep track of for proteins, so what about the chemical stuff? Well, in terms of elements, we still see carbon, hydrogen, and oxygen, just like we saw with your carbs and your lipids, but now we've got two additional enzyme elements to keep track of, and that's your nitrogen and your sulfur. So a couple of compound name endings to also be aware of with these. We saw that OSE tells me carbohydrates, but there's some that you may show up in protein names, and it can kind of give you a hint as to what you're dealing with. If it happens to end in IN, then we are often talking about structural proteins. If I back up a slide, right here is actin, myosin. I mentioned keratin in your hair and nails, and a lot of this is structural stuff. ASEs, these are your enzymes, and this is going to be pretty much consistent across the board. So anytime you see anything that ends in ASE, you're talking about some sort of enzyme. So definitely a name ending you want to memorize, because as you're coming across new words, You may not know what it is, but if you know it ends in ASE, then you know it's an enzyme and you know its basic function, even if you don't know specifically what that enzyme does. So just some more examples. There's the collagen, lactase, muscle, right? These are all examples of proteins. So let's get into monomer-polymer structure, because we're back to a class that does in fact have that. So when we talk about monomers or building blocks for your proteins, these are these guys called amino acids. And here's your basic amino acid structure, and I believe you've got to label this in your notes, so make sure you label the picture. You've got 20 different types of amino acids. Alright? And that's because if I look at this amino acid structure, the actual parts of that structure, there's some of it that stays the same and some of it that will actually change from one amino acid to the next. So if we take a look at those parts, every amino acid has this asymmetrical carbon. Asymmetrical, hopefully you know what that means, right? If something is symmetrical, it's the same on both sides. So this carbon is asymmetrical because it's got different things no matter which direction I look. So what are those different things? Well, here's your amino group, one of those functional groups I told you to know. You've also got a carboxyl group on the other side. So remember I mentioned this to you when we were going over functional groups. Why do we call it an amino acid? Well, there's the amino part, and carboxyl groups are acidic, so there's the acid part, so hence the amino acid, right? So I've got an asymmetrical carbon, an amino group, a carboxyl group. I also have this hydrogen that always comes off, and carbon's able to make 4 bonds, so that 4th bond is what we talk about as an amino acid side group, or R group. And this is what's different. Alright? So I've got different arrangements of elements that are coming off of that central carbon, and that's what makes the 20 different amino acids. It determines their property. And we're going to talk about protein folding today, and it's going to ultimately be those side groups that determine how our protein actually folds up. Put amino acids together in a chain and I'll eventually get a functional protein. So just a chain of amino acids are going to come together. It's going to be the OH from your carboxyl group of one amino acid minus an H off the amino group. Off's going to come your water, and we're going to have this blank bond between this carbon and this nitrogen to plug together. And that's what you're looking at here once this reaction, dehydration synthesis process finishes. And just like every category of macromolecule we've been talking about, there's a name for that specific type of covalent bond. In this case, we talk about them as peptide bonds. Alright. So here's the interesting thing about proteins. Great, I can make this string of amino acids, I can make this huge polypeptide chain, but the problem is that it's not a functional protein yet. Just having a string of amino acids is not enough to actually produce a functional protein. A protein that actually does some sort of job in your cells has to fold and twist and create this unique shape. This is actually a functional protein. Alright, so you've got all these twists and bends and curves, and that unique 3D structure is what actually gets a protein to perform its job. Alright, now how do I get to that point? Well, my amino acid sequence is going to determine where those twists go, where do those folds happen, and how the shape actually takes form. Okay? And really, ultimately, we're going to talk later this year that DNA is responsible for the sequence of amino acids, which then dictates how this protein forms. So really, it all is coded for in your genetics. So proteins have 4 levels of folding. Think of it like making one of those origami swans. If you don't do the right folds in the right order, then you end up with this jumbled piece of paper as opposed to this pretty swan at the end. Well, that's kind of like what's happening with your proteins. So let's talk through each of these levels. So if we start at the beginning, we're talking just your primary structure. And these are what amino acids are in your polypeptide chain. Alright, so which side groups are coming off of each of these amino acids, and in what order are those amino acids showing up. Alright, so this is your straight chain. This is ultimately what's going to dictate all the other levels of protein folding, because it's going to be those interactions then in those side groups that determine how the rest of this folding happens. So secondary structure comes next. Once we take a look at your protein amino acid sequence, then those side chains start hydrogen bonding with each other and either make these alpha helices, so these twists, or they make these beta-pleated sheets. So sort of think of them like if you've ever folded up one of those paper fans as a kid, that's kind of what these beta-pleated sheets look like. And in the string of amino acids, let's say I've got 100 amino acids, I may have from amino acid 3 to 20 an alpha helix, from 25 to 60 a beta-pleated sheet, and then from 60 to 70 I have another alpha helices, so you can get both of these in a single secondary structure from a single polypeptide chain. Just be aware of that. Tertiary structure then, alright, is going to kind of end up looking like this jumbled mess. So we get all of those alpha helices and beta-pleated sheets, and this whole thing kind of twists and bends onto itself and creates that true 3D structure. And this can happen really from a multitude of different types of interactions that are now happening between those R groups in your amino acids. So I can have some more hydrogen bonding going on. Sometimes there's ionic bonds going on if those side groups have some sort of charge to them. Those sulfohydryl groups, those sulfur hydrogen functional groups that we took a look at, they can actually find each other, join up, and create these things called dual sulfide bridges. So literally the sulfurs will bond to each other. Also, something we can add to this, hydrophilic, hydrophobic interactions. I think sometimes those are the easier ones for kids to recognize, because if I've got this 3D structure going on here, there's an inside and an outside to that protein. So if I have a amino acid with a side group who is hydrophilic, right, so it likes water, it's more likely to be found on the outside of this protein, because proteins are being made in your cells, and your cells are full of water. Flip side to that, if it's hydrophobic and that side chain doesn't like interacting with water, then you're probably going to find it buried somewhere deep here on the inside of that protein structure so that it can kind of get shielded and hidden from that water. Now for some proteins, tertiary structure, we're done. Alright, this is its functional shape. But in other cases, sometimes that tertiary structure really is just a subunit. And instead I've got to go one level higher, all the way up to a quaternary structure, in which case I'm taking those subunits and linking them together and making something like this. Now sometimes it's the same subunit, like you see here, so there's 4 of the same. Sometimes it's different subunits, sometimes there's 2 and 2. Hemoglobin is an example of a protein that makes it to the quaternary structure level, and remember, hemoglobin's what's carrying oxygen around your body as part of your red blood cells. Hemoglobin has an alpha subunit and a beta subunit, but it's got 2 of each, so it's got 4 total subunits, but 2 of them are alphas, 2 of them are betas. So really there's a whole bunch of different combinations and options here. This is just one particular example. So if I go back through, right, here's my amino acid structure, that's primary. Secondary structure, I start bending into these alpha helices and beta-pleated sheets, and the hydrogen bonds are what's holding those together. Tertiary structure, now all of that starts bending and coiling and folding back on itself, and in some cases this is the end of the line, this is a functional protein. In other cases, this is only a subunit that will then come together with other similar or identical subunits and ultimately make your quaternary structure. So your proteins are somewhere functionally here or here, depending on the type of protein you're talking about. So why is protein shape so important to living things? Well, it's going to come back to homeostasis again. So in order for the protein to do whatever job it is that it does, that protein's shape has to be present. But those bonds that are holding those protein shapes together are very sensitive to changes in the environment. So changes in pH, changes in salt concentrations, changes in temperature can all make those relatively weak-ish bonds that are holding those amino acids together in that fold to come apart. All right. So anytime that a protein has a situation where it finds its environment has shifted enough that it's lost its functional shape, it can't do its job anymore because of one of those altered conditions, we talk about it as a denatured protein. So here's a normal functioning protein, and let's just say you're running a really high fever, right? So what ends up happening, while your body is naturally trying to get rid of a bacteria or a virus, which, oh, by the way, are also made up of a lot of proteins, so we're ratcheting up the temperature, and the idea there is that your body's going to get the temperature high enough to denature the proteins or get them to unravel, in which case they can't do their job. If they unravel in that virus or that bacteria, then hopefully your body is able to then fight off and destroy that pathogen. Problem is that sometimes that temperature gets too high. So we talked about feedback loops in Unit 1, and running a fever is actually a positive feedback. So long as those bacteria or viruses, proteins are continuing to function well enough and not denature, your body will turn your temperature up. If it still doesn't work, it's going to turn the temperature up again. If you're still sick, it's going to turn the temperature up again. And the problem is, once you get into that range of like 103, 105, we're really in danger of now denaturing your own proteins. And that's where we start worrying about things like brain damage, because we're denaturing the proteins in your brain, which aren't doing their job. We're denaturing things like the hemoglobin in your body, which now can't carry oxygen to your cells. So that's one of the problems, that it becomes such a danger to run such a high fever. Alright, but initially the actual process is there to denature the proteins of the thing making you sick, which is kind of interesting.